Welcome to the Fantastic Four. There are four biomolecules: lipids, proteins, carbohydrates, and nucleic acids. First, we have lipids containing CHNO. The monomers for lipids are glycerol and fatty acids. One glycerol and three fatty acids bond to form triglyceride. When a phosphate group is added, it forms phospholipids. Fatty acids with double bonds form unsaturated fats, which are liquid at room temperature, and ones that have only single bonds form saturated fats, which are solid at room temperature.
Examples include steroids and fats such as seal blubber.
Second, we have proteins containing CHO and N. The monomers of proteins are amino acids which vary by their R groups. They bond together through peptide bonds, creating polypeptide chains. Examples include hemoglobin in blood and actin in muscle proteins. Third, we have carbohydrates containing CHNO. The monomers for carbs are called monosaccharides, and they include glucose and fructose. Two monosaccharides together are called disaccharides, which include sucrose, maltose, and lactose. Polysaccharides are long chains of monosaccharides, which include starch, cellulose, glycogen, and chitin. Lastly, we have nucleic acids, which contain CHON and P. The monomers of nucleic acids are nucleotides. Nucleotides are made of a phosphate group, a 5-carbon sugar, and a nitrogen base. The two polymers are DNA and RNA. Now we will look at how these biomolecules bond together and how they are broken apart. So we have two amino acids, and all of these orange looking colors represent hydrogen. So when these two amino acids want to bond together, this hydrogen from the amino group and the hydroxyl group from the other amino acid will bond together to create water. And this leaves this amino acid with a carbon that has an incomplete valence shell, because carbon has 4 bonds when it has a complete valence shell, and this nitrogen needs 3 bonds to complete its valence shell. So then these two amino acids will bond to complete both needs of the two atoms, and now these two amino acids have bonded, and this is called dehydration synthesis. And dehydration synthesis happens when an organism forms polymers with individual monomers, and an example is when a plant makes starches to store energy. Hydrolysis is the opposite of a dehydration reaction. The polymer is broken down into monomers by the addition of water. So now we have this water molecule and these two amino acids that have been bonded by the previous dehydration synthesis. And now when we add this water, so this hydrogen atom will then bond to this amino acid, this amino group, to the nitrogen, and this creates one amino acid, and this hydroxyl group would then bond to the carbon atom, creating another amino acid, which breaks this polypeptide into 2 different amino acids. And this happens when an organism consumes a polymer and needs to break it down in order to bring the individual monomers to its cells. And in this case, it brings these amino acids to its cells. And an example of this is during digestion, when a human breaks down carbohydrates from bread into glucose to use as energy.